Hello, everyone. Our lesson for today is about microbial cultivation and growth. So, in this lesson, you will learn how microorganisms are classified based on their nutritional requirements and how they acquire nutrients from the environment and use this for their growth. So, topics also on culturing microorganisms and measuring their growth will be discussed in this lesson. So, at the end of this lesson, you will be able to identify the different nutritional types of microorganisms, describe the process of nutrient acquisition by microorganisms, describe the process of microbial growth, describe the process of how microorganisms respond to stress and starvation. Describe the process of cultivating microorganisms and practice computing mathematical expressions of microbial growth. Okay. So what are the different nutritional types of microorganisms? Okay. First are what we call the photoautotrophs. So the photoautotrophs are those that use carbon dioxide as carbon source, okay, and light as a source of energy. Their electron donors could be H2, H2S, or water. Okay, so an example of a photoautotrophic microorganism is a bacteria called Anabaina. No? This is a culture of Anabaina that is grown the presence of carbon dioxide and light. So majority of these groups, no, or the nutritional types of microorganisms are actually classified based on their carbon source and their source of energy. So this is for the photoautotrophs. Another groups are called the photoheterotrophs. Photoheterotrophic microorganisms use organic compounds as carbon source, and just like photoautotrophic organisms, they use light as source of energy. They could use H2 or some organic molecule as source of their electron donors. Now, electrons, as we go to our metabolism topics later on, electrons are used for during the production of organic compounds. So the source of these electrons is really important for microorganisms. So an example of a photoheterotrophic bacteria is what we call rhodomicrobium that is grown in the presence of acetate and light. Another group or another type, nutritional type of microorganisms are what we call the chemoautotrophs. So they use carbon dioxide as carbon source and they oxidize inorganic no, substrates as source of their energy. And their electron sources are those inorganic compounds. Okay, an example is a Theobacillus metabolis. It's grown in the presence of carbon dioxide and sulfide. So this is a sulfur oxidizing bacterium. The fourth type of nutritional of microorganisms are the chemoheterotrophs. So they are the ones that assimilate organic molecules both as carbon source and as source of energy. Example is the Micrococcus luteus that uses glucose as source of carbon and energy. So how do we uh, remember these different classifications? Okay, so there are generally four nutritional types of microorganisms: photoautotroph, chemoautotroph, photoheterotroph, and chemoheterotroph. Their names are actually based on the source of energy and their source of carbon. Okay, so at the start, you ask your question. What is the carbon source? Is it from another organism, no, like an organic carbon source? If yes, you then ask yourself, is the energy coming from light? Then, if the carbon source is from elsewhere and energy from light, then that is photoheterotrophic organism. But if the energy is from the inorganic oxidation, the oxidation of inorganic compounds, then no, that microorganisms is the chemoheterotroph. Now, if the carbon source is not from organic compounds, then you ask, what is the source of energy? So if it is from light, then that microorganism is a photoautotroph. If it is from the oxidation of inorganic compounds, then that is a chemoautotroph. Now you should be able to remember that if carbon dioxide is the source of carbon, then that is an autotrophic organism. If organic compounds or inorganic or other compounds, then that is heterotrophic organism. If light is the source of energy, that is a phototrophic organism. If compounds, inorganic compounds are the source of carbon, is the source of energy or source of carbon, then chemotrophic microorganisms. Okay, so this is again another representation of heterotrophy and autotrophy. So in autotrophy, there is accumulation of energy, okay, and compounds. It could be carbon or organic compounds, to produce an organic compound. In heterotrophy, they utilize organic compounds, in order to they could produce energy, through the oxidation of organic compounds. So, a very good example of autotrophic organisms are those plants. They use carbon dioxide and light to produce their own food and energy, while heterotrophic organisms are us, the humans, because we use organic compounds as a source of our energy. So this is another representation of the different nutritional types of microorganisms. So based on their energy source and carbon sources. Again, if the energy source is light, nadjud nay photo. Okay? If the energy source are chemical compounds, nanay chemo. If the carbon source is carbon dioxide, nanay auto. If carbon source is organic compounds, then nanay siya hetero. So, photo, light plus carbon dioxide, photo, autotrophic organism. Chemical compounds and carbon dioxide, chemo, autotrophic organisms. Light as source of energy and organic compounds as carbon source, photoheterotrophs. Chemical compounds as source of energy and carbon source, are organic compounds, and the organism are chemoheterotrophs. So I hope you should be able to remember those nutritional types of microorganisms. Now, in reality, there are groups of microorganisms that you cannot just place in a certain nutritional type, because what makes them actually very successful is their flexibility or their nutritional versatility. Now, if there are microorganisms that are strictly belonging to a certain group of nutritional requirement, then we call them obligate. Okay, so for example, An obligate photoautotrophic organism will grow only in the presence of carbon dioxide and light. And an obligate chemoautotroph will grow in the presence of carbon dioxide and ammonia or sulfur. However, often a phototroph can also be a photoheterotroph. There are microorganisms with that ability. Or a photoautotroph can also be a chemoheterotroph. So if that is the case, then we are talking about of facultative photoautotrophic microorganisms. Other example could be facultative aerobes that can grow with or without oxygen. So we'll be talking more of that when we go to the different environmental factors that affect microbial growth. Now, in terms of the uptake of nutrients, generally in real life, nutrients are in low concentration in the environment. And also, that we know that the cell membrane is a barrier to nutrient acquisition by these microorganisms. We also know that polar molecules cannot diffuse through the membrane freely because the membrane is non-polar. Okay? Although water can diffuse freely because they're very small molecules, as well as apolar or non-polar molecules. Okay? So these are the different considerations when we talk about the different uh, the ability of microorganisms to uptake nutrients. So last time we've already talked about a different type of transport. So there are passive and different type of active transport. So we know that in passive transport, there is no energy expenditure. The microorganisms does not spend energy in order to transport a certain substance in and out of the cell. Active transport system uses no energy. It could be through ATP or secondary active transport through ion gradient. But there are also what we call group translocation na mechanism. Okay, so first is the passive transport. So you already know about simple diffusion and you already know about facilitated diffusion. So in facilitated diffusion, there is binding of the substrate. Now, and that binding induces changes in the conformation of the transport protein. And this is the one that speeds up the diffusion because we all know that there is higher concentration of these solute outside the cell. By theory, they could easily go inside. However, they're big molecules, or they could be a non-polar, or they could be positively charged. So they cannot really freely pass through this, but they can with the use of transport proteins. Okay? So the substrate is metabolized in order to maintain the gradient. Okay, so this is what we call facilitated diffusion. So it helps solute move across the membrane from a region of high concentration to one of lower concentration. It does not use energy and cannot move a molecule against its gradient. So this is an example of a substance that is transported across the plasma membrane of certain bacteria through facilitated diffusion. So you have here a glycerol, and the transport protein is what we call the GLPF. Okay, so the glycerol, as the glycerol enters the GLF channel, then a conformational change in the GLPF opens the channel to the cytoplasm and glycerol enters the cell. So that is how facilitated diffusion takes place. Okay. Now an example of an active transport. An active transport that uses ATP, energy from ATP, is what we call the ABC transporters. ABC meaning ATP binding cassette. Okay, so in ABC transport, you have a solute and a solute binding protein. Okay, so in a solute binding protein with the solute, as they bind no? As its complex binds to the membrane transporter. Then, what happens is that the ATPase activity, meaning the ability to use energy from ATP, that's what we call the ATPase activity, of one of the component, not this one, the yellow component, signals the opening of the channel, no, which is the blue, okay, and the movement of the solute, okay, into the cell. So this is what we call an ATP binding cassette because it uses ATP to move a solute across the membrane. It needs a solute binding protein in order to accomplish that. Now there are what we call secondary active transporters that uses energy, no, from ion gradients. So this is also what we call ion driven transport. So there is utilization of energy to move a solute, and energy comes from the proton or cation gradient in the cell. So if there are more protons outside the cell, that creates a proton gradient, and that will enable the proton to be transported inside the cell. So together with the proton, no, and a certain solute, a certain nutrient could be taken up, no, by the cell. So what are these secondary active transporters? So they could be uniporters, symporters, or antiporters. So this is the an active transport, no, of ions, no, through the proton motive force. So there is a charge gradient, meaning there is difference, no, in the concentration of protons in and out of the cell. So outside of the cell, this proton is taken in. Okay. So these uniporters transport one substance in one direction, such as protons. A symporter, on the other hand, transports two different substances in the same direction, no, such as a lactose. Okay. And an antiport transports two different substances in opposite direction. So a proton is transported in this direction, and opposite to that are the sodium, no, out of the cell. So these are what we call secondary active transport. So this is what we call a co-transport, no, symport and antiport. Because this is another representation of this active transport, a co-transport through symport and antiport. Now the another way in order to transport the certain substances, for example sugars, which is a very good source of energy for some microorganism, is the group translocation system. Okay, so this is what we call the PTS, or the phosphotransferase system. So the phosphate, no, is donated, okay, through the into the PTS system from the phosphoenol pyruvate, or the PEP. So this PEP, as it donates the phosphate, the phosphate is taken in by this enzyme 1. The PEP becomes pyruvate. Enzyme 1 with the phosphate complex passes on the phosphate to HPR. Now the HPR it could be the substrates, no, are transformed by phosphorylation during the co-transport. So say for example in here, the phosphate is then the enzyme 2A and 2B will then be phosphorylated. Okay. Now, as the sugar is being transported, enzyme 2C will transport the substrate, no, it could be mannitol, glucose, or mannose, across the cell membrane. And enzyme 2B transfers the phosphate to the sugar that is transported by the cell by the 2C. So as more sugar is being transported in, okay, more phosphate is also taken in, no, by this enzyme to be attached into the sugars that are taken inside. So this is what we call group translocation. So translocation na through phosphotransferase. So there is transfer of phosphate. Another way for microorganisms to transport, say for example, nutrients, example, transport of iron is through the process of what we call high affinity siderophore mediated iron uptake. So in in the environment, iron is highly insoluble. That's why some bacteria will excrete, iron chelators. No, these are chelators are highly reactive compounds, no, so that they could take in these highly insoluble compounds from the environment. So these iron chelators are called siderophores. Okay, so these siderophores are recognized by receptors. Siderophore iron complex could be recognized by receptors and internalized by transport proteins. So, for example, in E. coli, now, the siderophores produced are what we call the enterobactin or the enterochilin. Okay. So, how does it work? So, a siderophore, the enterochilin in E. coli, will chelate the iron, which is highly insoluble. Because the chelator is very reactive, the iron will bind through it, and with the enterochilin iron complex, no, this will be transported to the cell, to the periplasm, okay, through an outer membrane called the FEP-A. So since that's the outer membrane, and you have here the inner membrane and the cell membrane, this is most probably a gram-negative bacteria. Of course, E. coli is a gram-negative bacteria. So as the enterochilin iron complex enters the periplasm, it will be transported into the cell by an ABC transporter that includes a periplasmic solute binding protein called the FEP-B and the integral membrane proteins called the FEP-C, which is the ATP, has ATPase activity, the FEP-D and the FEP-G. Okay, so if you notice here, this is all an ABC transport. As the iron enterochilin complex is transported to the periplasm, a solute binding protein now will transport it through this ABC transporter. Okay, so this is another representation of a high affinity siderophore mediated iron uptake. Now let us talk about microbial growth. So we know that these nutrients taken in from the environment are used by microorganisms for their growth. We've already talked last time that microorganisms, the growth in microorganisms is actually through the increase in the number of cells, no, rather than increase in the size of the cell. So how do they multiply? So they do this by binary transverse fission. Okay, so the chromosome is replicated first to produce two copies, and then the septum is formed in order to produce two daughter cells. So it's more like mitosis, but not really the same, but more like this mitosis in higher organisms. So the binary transverse fission is divided into two phases, the C phase and the D phase. So at C phase, the cell's genome undergoes replication to form two identical daughter DNAs. And at the D phase, the daughter DNAs separate in the process involving the cell membrane and will move to opposite ends of the elongating cell. Now, aside from binary transverse fission, some groups of microorganisms, they also reproduce via budding or polar growth. So, in both cases, a small protuberance or a bud, magbukol, forms on the mother cell and will enlarge as growth proceeds. When the bud is sufficiently mature, it separates from the mother cell. The mother cell, na in budding, produces another bud from the same location on the cell surface, and daughter cell produces the first bud. However, in polar growth, newly synthesized cellular components are distributed unequally between the two daughter cells. Now, when there is low concentration of nutrients in the environment, and the condition is not really conducive for their growth, so microorganisms become stressed and they become starved, no? Gutuman sila. Okay? And so this will affect the microbial growth. And so in order for them to still survive, okay, so what they opt to do is to cease, their metabolic processes, but still keeping their genetic materials intact. How do they do it? So some groups of microorganisms are capable of producing the what we call the resting cells, now what we call the endospores. Okay, so these endospores have resistant protein coat. We already talked about this last time. No, the formation of endospore is called sporulation. So this is most probably a bacillus uh, bacteria that produce endospores. So this is through Schaffer-Fulton stain. In the laboratory, you will do endospore staining also using malachite green. So the green here, now these are the spores, inside the cells. Okay? These endospores are now released. Now, in the laboratory, we could mimic an environment where it could be conducive for the growth of microorganisms. So we can actually culture microorganisms. So in culturing microorganisms, you should be able to provide all the necessary nutrients that the condition that is favorable for the growth of microorganisms. So the Shelford's law of tolerance states that abundance and distribution of organisms can be controlled by certain factors like temperature, the solid concentration or water activity, the pH, oxygen concentration, barometric pressure, and electromagnetic radiation, and many other factors. What we're talking here is that you should be able to understand your target microorganism in order for you to culture them. So, for example, in pH, you cannot culture an acidophile in a basic culture media. Okay? So you should be able to understand the biology, the physiology of your target microorganism so that you will be successful in culturing them. And no growth is a limiting resource for microorganisms. The Liebig's law of the minimum states that nutrients in least supply relative to bacterial needs will cap growth yield. Okay? So it is very important that you will be able to provide the necessary requirements in certain amounts so that even if usa sa mga major nutrients is missing, that would still affect the growth of the microorganisms. Now, in a closed culture system, what we mean closed culture system, so you have a petri dish. No, you already put all the nutrients in there and you put the bacteria. So you just wait until the cells will multiply. You don't add more nutrients and you wait until the nutrients will be exhausted. So that is what we call a closed culture system. Okay? Di na ka magsigdug ang nutrients. Hanurutan ni mga mahuruto siya. So how do microorganisms grow in a certain culture system? So they are measured in terms of the logarithmic numbers of bacterial cells. So logarithmic man meaning it's a base of 10. Exponentially, it's times 10. Okay? And the number of viable cells over time. No? So once you introduce, say for example, 100,000 cells in a particular petri dish, at a certain time, it will still not grow. Hindi pa sila mag-divide. Then, pag naka-adjust, they will still adjust to the environment. Then this is what we call the lag phase, the adjustment period. But if they're already adjusted and they found out that there are so many nutrients, then they will grow exponentially. So this is the log or the exponential growth phase. However, there will become a time that the nutrients will be limited and there will not be enough nutrients. So others will be dying. So the bacterial growth with the growth will now reach the stationary phase wherein the number of those dying are equal to the number of those new cells that are produced. And there comes a time when there will be no more nutrients. So all of them will start to die. So this is now what we call the death or the logarithmic decline. This is the logarithmic growth, nor the exponential growth. This is the logarithmic decline phase or the death phase. So you should be able to understand this phase, to be familiar with this phase of microbial growth in a closed culture system. So again, these are the four phases of microbial growth in a closed culture system. So the lag phase or the adjustment period, the exponential phase or the logarithmic or the exponential growth phase, the stationary phase, there is no growth, or the survival period of microorganisms, and the decline or the logarithmic decline phase. Okay. So again, the lag phase is a period of adjustment, no cell division, of course. Here in the logarithmic exponential increase, the exponential growth, the generation time or the time it takes for the cell to double is constant. So magsige na siya grow and grow. In the stationary phase, their production and cell death are exactly balanced. So there is no growth rate. When the death rate exceeds the rate of reproduction, the cell population is in the death or decline phase. Okay, now what happens during the stationary phase is that some of the cells will die or lie. So the cells become more resistant to adverse conditions. And this is the time when the cells also will synthesize their storage material. And different genes are expressed, producing secondary metabolites, needed for the survival of the organism. So in this closed culture system, how do we express microbial growth mathematically? So exponential growth phase is expressed by this equation. Okay, the final number of cells is equal to the initial number times 2 to the power of n times 2, because we know that it is exponential. It is doubling all the time. But how much double, we don't know. Okay? So, say for example, if one cell could yield 2 cell, this is the visual representation, 2 cell to that power. And so, the number of generations, the n, could be derived by getting the log of the final number of cells minus the log of the initial number of cells over log of 2. Okay. Well, the growth rate constant could be how constantly the bacterial cells are dividing. Okay, so the K is equal to the N, which is the number of generations over time. And this is expressed in units of generations per hour. So how many generations, kapila man siya mag-divide in a particular hour. Generation time, on the other hand, is the the time it takes for the cell to double. If one how in how many minutes can one cell become two? That is generation time. So G, the generation time, is equal to 1 over K. And this is expressed in units of hours. And we know that the K is the growth rate constant, which is the N over T. So the generation time is also equal to the doubling time. Thus, K is equal to 1. K is equal to the log of this. No, the over 0.301 times T, which is the constant. Okay, so you have here an example of a problem. So Escherichia coli divides every 20 minutes. You inoculate a culture, meaning you transfer or you replace a culture with exactly 1,000 bacterial cells. After 3 hours, how many bacteria are present? Okay. So the E. coli now divides every 20 minutes. Is that the generation time? Yes, that is the generation time. No, every 20 minutes it will divide. And you have here, you inoculate a culture with exactly 1,000 bacterial cells, meaning the initial, the N, no, the N 0 is 1,000. So after 3 hours, how many bacteria are present? So what it is? It is now asking about the final number of cells. So how will you solve for that? So given the initial number of bacterial cells is 1,000, therefore N0 is equal to 1,000. In 3 hours, E. coli will divide 9 times. So how did we get 9 times? No. Because in 3 hours, there are how many minutes? There are, in every hour, there are 60 minutes. So 60 times 3. Okay? And we know that it is every 20 minutes that the cells will divide. So for 3 hours, how many divisions had occurred? How many generation time has occurred? No? So that is what we call the generation time. So 30 times 60, because in minutes may siyang expression, so over 20, the generation time equals 9. Meaning, in 3 hours, E. coli will divide 9 times. So N is equal to 9. So using the formula NT, or the final number of cells, equals the initial number of cells times 2 to the N. You substitute the given initial number of cells and the number of generations, and this will give you NT is equal to 1,000 times 2 to the 9, which is equal to 512,000. Thus, it means there will be 512,000 E. coli cells that will be present in the culture after 3 hours. So that's how you will be able to solve for the uh, expression of microbial growth using this formula. Now, in cases, you will be given a certain values. So the very important thing is that you should remember the formula so that you can derive from them, because it depends on what is missing during in the problem that will be given to you during the exam or in the quiz. Okay, so that's all for the topic on microbial cultivation and growth. So in order for you to effectively review this lesson, you have here our summary questions or guide questions. So first, what are the nutritional types of microorganisms? What are the processes involved in the nutrient acquisition of microorganisms? How do microorganisms grow? How do microorganisms respond to stress and starvation? What are the different ways to cultivate microorganisms? And how can you express microbial growth mathematically? So if you are able to answer these summary questions, you will do very well in learning the things that we have discussed in this lesson. Okay? If you have questions, Alright, that our clarifications regarding our lesson. You just contact me through my email or the email of your lecturer. I also would like to acknowledge Miss Maria Shalita Escrsal for the help in making the slide presentation. Okay. Also, I would like to apologize because the recording software that I am using has been having problems. That's why I need to remove the video of me discussing, and instead just have my voice recording remain. Okay, but I will try to figure it out what happened, and so that I will be able to have my video in the next lesson. Okay, so thank you very much, and see you again in our next lessons.